Thank you so much for being here and bearing with us for the setup. So I'm Olivia. I'm just going to get us kicked off today. But all three of us are going to be talking about how we are using our graduate student research at Cal Poly Humboldt in the Department of Fisheries Biology to monitor some native fisheries here in Humboldt. So thank you again for being here, and thank you so much to friends there, Arcata Marsh, for hosting us. So where we're sitting right now, also Arcata, also Cal Poly Humboldt, as well as some study sites we will be talking about today, including the Yurok, the Eel River, the Bagwat, the Mad River, and Weehee Humboldt Bay. These are all located on the unceded ancestral territory of the Yurok peoples. So this is going to include the Yurok Tribe, the Bear River Rancheria, and the Blue Lake Rancheria. Some other study sites we will be mentioning today, Ari and Osage, are located on the unceded ancestral territory of the Yurok peoples. This includes the Pulikla Tribe of the Yurok People, the Trinidad Rancheria, the Yurok Tribe, and the Big Lagoon Rancheria. So these lands and these ecosystems have been stewarded by these tribes since time immemorial. The Wiyot and Yurok people continue to be an integral part of these landscapes and the research and the monitoring happening on the fish we're talking about today. And these species we're talking about today are of important cultural, livelihood, and ceremonial value to the tribes. This is just something to keep in mind as we talk about our fish today. So we are part of a larger project called the North Coast Tribal Fisheries Collaborative. This is a working group of tribal, academic, state, and federal partners where we have this shared goal of improving the climate resiliency of culturally and economically important tribal fisheries in far northern California. So there are 4 tribes supporting this project: the Blue Lake Rancheria, the Trinidad Rancheria, the Pulikla Tribe of the Yurok People, and Tuolumne Nation. So we are 3 of 5 graduate students on this project, specifically working with the Blue Lake Rancheria and the Pulikla Tribe of Yurok People. And with our specific project, some of our main overarching goals for our research is just to help increase the capacity for fisheries research within the tribes. We hope to do this by filling in some gaps in data and co-creating some new methodologies to monitor species of interest, like the green sturgeon, the Chinook salmon, and the surf smelt. So this here, I know these are a little small, but I just wanted to showcase all the graduate research being done under the collaborative. So like I said, we're 3 of 5 grad students. We are the 3 specific fish-focused projects, but when you include all 5, this research is ranging as far south as the Eel River and all the way up through northern California. So about how this presentation is going to go, I'm going to pass off in just a second to Daniel, who's going to come in and talk about a coastal species, the one here in blue, the surf smelt. Then Rachel will come in and talk about Chinook salmon, specifically in the eel. And then I will return and talk about green sturgeon in Humboldt Bay. So like Ava said, we would prefer questions at the end, but since we're giving like 3 different species talks, if there's any burning questions after each fish, we'll take a minute. But we prefer maybe to write it down to the end for the sake of time. And yes, I'm going to pass on to Daniel.
Hello everyone, 1 second, I'm just going to take this thing off the top. Hello everyone, my name is Daniel Montoya. Today I'm going to be talking about collaborating with tribal partners to co-produce knowledge on surf smelt populations across Humboldt County. So a little bit of background on species and question. So surf smelt or Hypomesis proteosus are data poor in our region. So the Tuolumne Nation has done a habitat assessment on them, and they still currently monitor their habitat. And then all of the peer-reviewed literature on surf smelt comes out of the Puget Sound. And then, so why study surf smelt in Humboldt County? First reason is they're ecologically important. Second, they're culturally important. Third, there's a small commercial and recreational fishery. And then lastly, it helps us fill in those data gaps. And then, so a little bit on their biology, they can be found from southeast California with some years sighting each. It's thought that they live up to 5 years old and reach maturity sometime between 1 and 3 years of age. They're iteropares, meaning they spawn multiple times in their lifetime. And it's thought that they spawn multiple times in a spawning season. And one thing I wanted to point out, I'm not sure anyone is familiar with the night smelt, but they are another family, or they're another species in the same family as Meridae. They share the same spawning habitat up in Humboldt County. spawn around the same season. The one difference is they spawn during the nighttime, while surf smelt spawn during the daytime. Really cool fact, the family smells like cucumbers for some reason. And then you can find them spawning on coarse sand and pebble beaches, looking for 1 to 7 millimeter size sediment. And then their sex ratios have been observed, very male skewed, up to 8 to 1. And then, so I wanted to quickly touch on their ecology. So they're right here in the middle. They're going to be forage fish, and they feed on plankton such as amphipods, copepods, as well as marine worms. And they pass that energy up the food web to larger predators such as coho and Chinook salmon, and marine mammals, marine birds, and humans as well. And then, so I just want to touch on the cultural importance of surf smelt. So indigenous communities in the Northern California coast have been feeding on them since time immemorial. And how they do that is through the use of this net. So this is a traditional A-frame net. And what these groups would do is they would look for building materials around them, make these nets each year, and then fish in the surf when the fish are spawning. And so when they catch the fish, women like these would take them and dry them out, customary on dune grass. And this is another photo showing the smelt camps. So this is up in Crescent City with the Tala Dene Nation. And so there's actually 2 families that still actively fish surf smelt each year. And so they go stay on the beach for a couple weeks, dry out their fish, and they get to enjoy this throughout the year. And so you might be wondering, is there a number to time immemorial how long they've been eating these fish? So one really cool study, it was looking at archaeological sites around Northern California and Southern Oregon, and it was really cool. They found a lot of fish bones, and specifically in Humboldt and Delmar County, they observed mass smelt harvesting. And so the left figure is Del Norte, and then the right one is Humboldt County. And so you can see that it's very smelt dominated, with Del Norte comprising, or smelt comprising 92.6% of the findings at that site in Del Norte, and then 86.5 in Humboldt. And so I'm not saying it's all they ate year round, but at these sites it was really big. So there was like evidence that these groups were feeding on them for a long time. And then, so to touch on the commercial fishery, this figure on the left shows landings by participation. On the left, we have the landings. On the right, we have participation. The landings are going to be the blue blocks. The participation is going to be in orange. So if you look where that arrow is, you can see a sharp decline. And what that is 1997, the Redwood National and State Park actually implemented a permit system for the commercial smelt fishers. And so in order to access the beach with your truck, which is how you commercial smelt fish, you need a permit. So that greatly brought down participation, which would then bring down landings. Not as many people fishing, not as many fish being caught. And this photo on the right shows just kind of what it looks like to commercially smelt fish. And then, so I'm going to touch on my objectives of my study. So the first objective is to co-produce a long-term study on surf smelt with the Pulikla Tribe of Yurok People. So this is their land. This is the region up here. They're just south of the Klamath River. They were previously known as the Blue Rancheria. And then my second objective is to study the population by measuring the length, the weight, sex, and age of spawning individuals. And so how do we do that? Well, what we do is, it's really fun. We get to go to the beach and we are either going to walk or drive looking for ecological indicators. And then what we do is we're going to sample for 1 to 2 hours with one of those traditional A-frames. And it's really nice. This is our net in this photo. And so my collaborator, Tyler Jones, he hand built the nets. And so what we do, we're going to keep the first 30 fish to process later. And then we just count and release everything after that. And then we're collecting physical data, such as visual assessments of weather. Is it sunny, cloudy, things like that? Kind of helps us with what were the conditions like and how can we see like ecological indicators, things like that. Then we're estimating wave height and looking at spatial data, such as the distance that we sampled, how far did we drive that day or walk that day, and where did we specifically catch those smelt that day. And then with the 30 that we keep, we take those, we go to the Telonica Marine Lab, which you can see up top. And then we're collecting fork length on them, total length, weight in grams, and then we're sexing them, and then we're removing the otoliths. I'm not sure if you've heard of them, ear bones. It's this photo to the right, similar to trees. They have annual readings that you can count and age them through. And then we're also collecting their stomachs and preserving them to see what their diets are at a later date. And then so to get into my sites, my southernmost site is Centerville, then I have Mad River Beach, Redwood Creek Beach, and Gold Bluffs Beach. Centerville and Mad River are sampled once a month, and then Redwood Creek and Gold Bluffs Beach are sampled twice a month, and they're sampled on consecutive days around lunar phases. And then I want to touch on some of those ecological indicators that I mentioned. This is a fun way for the crowd, if you're out at the beach this summer, to find some smelt. This is how to do it. So first, what we see are brown pelicans, and we see those kind of far offshore. They dive bomb, so they can be anywhere from the furthest breaker to the first breaker. They generally tell us how far in the surf zone they are. And then we see double crested cormorants. They're going to be sitting on the top of the water, and they'll dive down, catch smelt, come up. We can see smelt in the mouth and kind of tell also where in the surf zone they are. And then we'll see harbor seals, and they pass through the closest breaker, generally feeding on smelt. And then this is kind of the best one for us. It tells us when to fish. So seagulls, they're going to be on shore. And once they approach the water, then there's smelt running and it's a good time to fish, good area to fish, things like that. And then something else, we see California sea lions as well in the same area that we see harbor seals. And then we see surf scoters diving offshore, and we see ospreys working in the surf zone as well. And so just to touch on co-production, how do we accomplish that? Well, it started by the tribal partners and Cal Poly staff selecting the sites, and then we discussed what parameters we'd like to collect throughout the season, and then we jointly sample for surf smelt together. And this video, it was really cool. It was showing these ecological indicators. I just want to go to one clip for a second. Just pretend you saw a bunch of birds and but to keep going on our co-production, so the tribal partners and I, we continued by processing those samples, and then any of the documents are approved by the tribe and their natural resource team. And then we're going to communicate and make ongoing adjustments as needed if we need to change any collection methods, improve the project, things like that. And then, so these are our smelt catches. I'm going to be starting off with Centerville. So on the left, you can see the dates, and on the bottom, you can see the counts. So you can see that in June, we caught our first fish at Centerville with 6 fish, and then it rose up to 108. And then in August, we caught 30 fish. So pretty variable. And then if you look at Redwood Creek, pretty interesting. We caught a fish in March, and then we caught some in June. That site was really interesting because once the river mouth closed, the fish stopped spawning, and we stopped seeing those indicators and presence of smelt. And then Gold Bluffs, pretty interesting. In June, we caught some fish, and then in August, we got 1,200. So it's highly variable. And my fourth site in Mad River Beach, we didn't catch any fish. And most of our catches were through March through October. And then so looking at some lengths of smelt, I have total length on the left and then sex on the bottom. And it's showing the three different beaches. So Centerville on the left, Gold Bluffs in the middle, and Redwood Creek on the right. And what you can see is that the males are going to be longer than females in gold. And then this is just a different way to visualize that, where it's kind of displaying how many we catch at Gold Bluffs. And then you can see that there are some bigger ones past the 170 mark, where you don't see in the females. And then, so touching on the smelt weights. So on we did have some, oh, sorry, excuse me. We had some heavier females over here at Centerville and Gold Bluffs, but at Redwood Creek, we didn't have any. And then same thing, you can just see the counts over here on the left and then weights down here. And then, sorry, males on the bottom, females on the top. And then looking at the sex ratios, so this is Centerville, and you can see females are counts are going to be on the left, sex on the bottom. So females are going to be in orange, males are in blue. What you do see at Centerville is a lot more males to females, and this is a common trend. Redwood Creek, we had no females that we processed. And then Gold Bluffs, we had same thing, a lot of males. And then, so in summary, this is the first study of smelt biology in Humboldt County. And then by working side by side with my collaborators, it allowed them to rediscover a common resource and improve co-learning experience. And then smelt were present from March to October at 3 of my 4 sites, but mostly peaked from June, between June and August. And then on average, males were longer, but females were heavier. And then sex ratios were heavily biased for and it sets the groundwork for the tribe to monitor species long term for continuing to things along to Rachel.
Okay, hello, everybody. Thank you for sticking with us. My name is Rachel Hein. I'm a graduate student in the Fisheries Department, and my advisor is in water. But I will be speaking on my graduate research, which is messaging how to manage Chinook salmon utilize the Eel River estuary or the So we probably are familiar with the Eel River estuary here in Humboldt County, but the Eel River itself.
Change.
That's the
If the guy had just talked, I would have thought that it's not.
Any different habitats that are available for fish species to use. So we have some cold water at headwaters and tributaries higher up in the system. We have warmer but variable mainstem habitats, and where the river meets the ocean, we have the Eel River estuary, which is where I will be focusing my talk today. And I just wanted to introduce you to some of the anadromous species that are in the eel that are not Chinook salmon. If you don't know what anadromy means, it refers to species that are born in freshwater, out migrate to the ocean to grow, mature, and then come back to freshwater to spawn. So we have all of our salmon species right here, steelhead on the top, coho in the middle, and Chinook on the bottom. But we also get green sturgeon and occasionally white sturgeon in the eel. Pacific lamprey is actually the namesake of the Eel River. And bottom here we have Yurok, which is a culturally significant species and historically it's been in the Eel River, but is thought to be extirpated. And in the middle we have American shad, which is our one non-native anadromous species, which utilizes the eel. So I just will quickly touch on the salmon life cycle. I'm sure most of you are familiar with this, but as I mentioned, the salmon are anadromous. So we're going to start with the adults here in freshwater. The adults will create nests called redds, which they will lay their eggs in. Those eggs will then mature into alvin, these little cute guys right here, and they have yolk sacs that fortify them for the first few days of life. Those alvin then transition to fry. The fry can turn to grow into par, and par will undergo this process called smoltification, where they will prepare to enter into the ocean part of the phase and go through physiological changes to incorporate all of that salt water. Then they enter into the ocean, where they usually spend between 1 to 4 years before they return as adults to spawn in freshwater. So for my work, I'm focusing on what I'm doing the estuary phase, or the sort of intermediate phase between the freshwater and the marine phase. And the estuary phase is an important intermediate step for these fish, as it allows them to transition from those par to smolt and go through that smoltification process. And the estuary itself will harbor all 3 of these life stages at one point: fry, par, and smolts. So now to introduce you to my study species, which is the Chinook salmon. You may have heard this salmon be referred to as the king salmon, but Chinook salmon are the largest salmon species. In the Eel River, they have typically fall-run species, which means they enter back into the eel in early fall and then spawn in fall and then into early winter. The Eel River Chinook salmon are part of the California Coastal Evolutionary Significant Unit, which includes Chinook salmon which spawn from Redwood Creek in the north down to the Russian River in the south. And fish that fall within this category are federally listed as threatened under the Endangered Species Act. Unlike some of the other salmon species, Chinook salmon are thought to be estuary dependent, which means they spend some part of that stage rearing or growing and maturing in the estuary. And this can be beneficial if you are a salmon species because there's higher foraging in the estuary, allows you to acclimate to those higher salinic areas, go through that smoltification process, and you can escape some of the conditions that are adverse, as warming temperatures or freshwater predators. So the Eel River salmon have experienced drastic population decline, upwards of 95%. We know that historic run for estimated to be up to almost 800,000 individuals that were returned annually. That is from cannery records for canneries that existed in the Eel River estuary at that time. Present day, there's thought to be less than 20,000 that return annually each year. And this is largely attributed to the combined effects of logging, overfishing, habitat degradation, invasive species introductions, dams, and water diversion projects. In the Eel River estuary in particular, there has been a few reports on juvenile Chinook salmon. The last occurred in 1995, and previous to that, there was one in 1977. So in the estuary itself, the habitat has undergone pretty large changes that occurred at the late 18th century when European settlers moved into the area. They began building these levee systems, as you can see in this image, which essentially protected farmland and agricultural areas from flood inundation. By doing so, they severely limited the amount of habitat that's available for salmon species as well as other sensitive fish in the estuary. So recently, there's been a movement to create these restoration projects that return sort of the natural regime of these areas and bring back in that natural tidal connectivity patterns and create more habitat in the estuary for these fish species. So this image here is actually from a restoration project that completed pretty recently. It's called Ocean Ranch. It's in the northern part of the Eel River estuary. And they're essentially breaching this levee here, returning tidal flow back into the area. So since 1995, there's been some pretty large changes in the Eel River estuary. There's been some positive changes, such as restoration projects. There's 2 large-scale restoration projects here in orange that were both completed within the last decade. And in addition, there's 2 restoration projects here in red that are planned to occur within the next few years. On the not so positive side of things, we also have this Sacramento pike minnow. There's an invasive cyprinid. Sacramento pike minnow are native to other regions of California, but not to the Eel River. So this fish was introduced in the 1970s, and it has since expanded to every single habitat in the Eel River, which includes the estuary. And this is a problem for salmon species because this fish is a generalist predator and is known to predate on small fish and even salmon species. So then for my project, these are the guiding questions that shaped the research that I'm conducting. So one, are juvenile Chinook salmon utilizing the Eel River estuary? If so, how are they distributed on the landscape spatially and temporally or through time? And what factors might be influencing those observed distributions? So I first had to come up with this way to classify estuary habitat. So if we think of this as a very simplified diagram of what an estuary might look like, the largest area here would represent the main stem habitats. This would be main stem Eel River, which I refer to as primary habitat. Then we have these secondary sloughs that branch off that main stem habitat, which I'll refer to as secondary habitat. And then coming off of those sloughs, we have the wetland channels that usually end blindly, and those will be referred to as tertiary habitat. So here is the Eel River estuary. It's a very large area. Essentially, it spreads from Ferndale down in the south here to Table Bluff and Pulikla in the east. And this is a very large area to sample, pretty intimidating. So in a way to break it up, I chose to split this into 7 different regions based off those habitat classifications I presented in the previous slide. So these 3 regions here, Crab Park is nearest the estuary mouth. So this is like the saltiest that a site would get. Cockcraven Island is in this middle estuary region. And Fern Bridge is at the top, which is essentially the top end of the estuary. I then have 3 sites that incorporate the southern slough channels and Salt River, the middle estuary, Kinawa Island, and up top, the McNulty Slough. And for tertiary habitat, we have this one site, oceanic, that restoration. So this is just a schematic. All the sites I have, you can see they're very spread out throughout the estuary, but just to give an idea of what some of this habitat looks like. This here is a tertiary site, so this is sort of a pond structure at the end of one of those tertiary channels up in the restoration project. We have some more area that is more on the riparian side down in the Salt River. And then there's a few pictures here of that primary habitat, so very wide channels. And how do we actually sample for these fish? So we use seine nets, which you may or may not be familiar with, but depending on the habitat, we'll either use a large seine and put it on kayak, or we'll use a hand seine like this. Essentially, they work the same way. You put them out in the habitat, you pull them in, get all your fish out of there, speciate and count up everything you have. Of course, we're looking for juvenile Chinook salmon, so if you're giving me those guys, we will measure and weigh them. And I'm also taking water quality measurements to try to get at that question of what parameters are influencing the distribution of this fish. So I will take dissolved oxygen, temperature, and salinity, and depth for every sampling we do. So now I'd like to touch on some results from my first sampling season. So I began sampling in the summer of 2024. I started very briefly in March and April, and then did the whole sampling suite from May through August. So I first just want to touch on the species diversity we found in the estuary. In total, we documented 21 different species. Using the Eel River estuary, it needs to be broken up into 3 different categories. You have your migrants or indigenous fish that will come in and use the estuary very briefly, maybe for spawning or rearing. You have your residents, which include things like sculpins and your gobies, and then your freshwater species, which are found higher up in the estuary, stuff like pike minnow and things of that sort. So just to show what some of these look like, I don't stare at fish all day, but you guys might want to too. But we have 2 species of smelt that we capture. This one here is longfin smelt, which is actually a threatened species on the California endangered species list, so it's cool to get that guy. Surf smelt, some species that Daniel just finished talking about. Also get them in the Eel River estuary. This guy here is a saddleback gunnel, pretty cool looking snake-like fish. My favorite are these bay pipefish, which are actually related to seahorses. And for our flatfish category, we have this lovely starry flounder. So this diagram here is showing the top 18 species we captured over the 2024 season. We did capture more species than this, but they were less than 10 individuals in total, so they did not make it into the top 18. But what we can see from this is that our least captured individual species was actually our target species, so Chinook salmon. And the species we captured the most often fell into the silver size category, which incorporates jack smelt and top smelt. And this included both adults, you can see here in the blue, but also a ton of juvenile fish that were rearing. So now I want to return to my questions and see if we can get some answers. So the first one, if you remember, is are juvenile Chinook utilizing the Eel River estuary? And this figure here shows my sampling regions and each of the months we sampled last year. If there's an X, it means that I did not sample that area. If there's a dash, and it was sampled, but we didn't find any Chinook salmon. So what we can see is that we found Chinook salmon from March through July of last summer, and there was a definite peak in June where we captured the most individuals. But overall, we documented 25 juvenile salmonids or juvenile Chinook over the 2024 season, so very low numbers. But now that we know they are using the estuary, we can ask those secondary questions. How are they distributed on the landscape spatially and temporally? So first, I'd like to answer the temporal aspect of this question. So this figure here is the flow of the Eel River and the fork length of our captured juvenile Chinook salmon. Basically, all you need to realize is that these peaks indicate large storm events. And if we map on what our juvenile salmon look like at this point, we can see that we have both fry and smolts that were utilizing the estuary throughout this period. We had fry come in early in March, and it's interesting to note that we have this cluster of individuals show up after a large storm event, which suggests that the water flow might be impacting their immigration down to the estuary. So now we'll get at that spatial aspect. So this figure here is showing where we captured Chinook over the course of the 2024 season. And the size of the circle indicates the abundance in that area that we saw. So the bigger the circle, the more fish we caught there. What we can see is that they're primarily located in that main stem habitat. But if we break it down by season, it comes to be a bit different story. So this first figure here is March and April, second, May, June, bottom is July. And if we map on our habitat classifications, it becomes evident that they were using that tertiary and secondary habitat earlier in the spring and transitioned to majority primary habitat later in the summer. So this begs the question, what is influencing those distributions? In part of my research, I also documented the summer temperature throughout the estuary. So each of these dots on here are essentially temp loggers that I had out through June through August. And this indicates the mean temperature for each logger. So just briefly, what we notice is that closer to the estuary mouth, things tend to be colder. And as you branch out from the estuary mouth, things warm up quite quickly. So if we map on our juvenile Chinook salmon, we can see that they are just using that primary habitat, and they seem to be using those lower regions, which suggests that there may be some sort of influence of temperature during the summertime, and it's really constricting the areas that they might be able to use. So I just wanted to end with a quick update. So this year I started sampling in February, and I'm sampling up until September. And what we can see, which is different from last year, is that we actually have captured a lot more juvenile Chinook this year than we did previously, which is very interesting. But I also want to note that We didn't really get out and sample a whole lot in March and April last year, so we could have been missing a lot of these same individual sized fish that were there. But an interesting pattern, so I'm curious to see if we get more individual trout the rest of the summer. But all of those, or a lot of those little fish that we captured in March and April were actually this size, and these are little fries. So it's interesting to see the majority of that migration be those smaller fish. And something that was also different, so we found a lot more coho utilizing the estuary in the winter time. And we documented different life stages as well. So both of these fish were found the same month within the estuary. This is a coho fry here, coho smolt. And these are actually also a year apart, which is very interesting. So that sort of has prompted us to expand the study to now include coho as well. And I just wanted to end by giving a shout out to my field crew, because this is pretty difficult work and it takes a lot of hours schlepping through the mud out there. So I couldn't have done it without them. And with that, pass it off to Olivia.
Any burning questions before I? Fantastic. Great. So I'm gonna shift gears one more time. This is area. The green sturgeon. And I'm just talking about how I monitor their presence. Oh, it's good. So first I want to take a step back. When I say take a step back, I mean millions of years ago. So this on the right is present day. A little to the left is we start having records of human fossils 100,000 years ago. And then I've left 3 blanks: 6 million years ago, 65 million years ago, and 200 million years ago. I'm wondering if anybody has a guess where I'm about to put a sturgeon on this. Interesting. So 6 million years ago, we have fossil records dating back for some salmonids. They think likely the Chinook salmon, which we just talked about. 65 million years ago, we have fossil records dating back for the T-Rex. And 200 million years ago, even like 230 million years ago, we have fossil records dating back for the sturgeon, including the green sturgeon. If there's any dinosaur buffs, this is the Triassic period. Yeah, do that what you will. Age of the dinosaurs, right? Older than our T-Rex. So often sturgeon, including green sturgeon, are referred to as living fossils. They are called this given that they haven't changed very much given that time frame of 200 million years. What they are thinking is that these dinosaur sturgeon ancestors, the main difference is they had a like bony skeleton like humans do, where the sturgeon we see today are mostly made up of cartilage, but they've still held on to some of this bone. They've got some in the heads, they've got some in the tail, but the most dinosaur-like feature are these scutes. This is a close-up picture. These are bony armored plates, so these just essentially act as armor. They have 5 rows, 1 on the top, 2 on each side, then 2 kind of adjacent on the bottom. And I like to imagine this is just a dinosaur feature they've carried with them. So this here is the 27 species of sturgeon in the family Accipenteridae. We have our green sturgeon. And I want to point out this white sturgeon here. This is our other native sturgeon species on the west coast. A little bit different in their ranges, but they are here in California as well. And these are the only 2 we have out here. But all 27 of these species are listed on the International Union of Conservation for Nature, so the IUCN Red List, if you're familiar. They are either listed as vulnerable, endangered, critically endangered, extinct in the wild, or just extinct. So sturgeon are often referred to as one of the most and hopefully use some of this information kind of study as to why they're so vulnerable. So we have a dinosaur fish that comes with a kind of vulnerable life history. This includes a long-lived lifespan. So green sturgeon can live around 60 to 70 years. The white sturgeon are other sturgeon species out here. There's currently some fish in British Columbia that are over 100 years old. And with this comes this late to mature nature. So green sturgeon, like a lot of other sturgeon species, don't mature until they're about 15 years old. In the context of fish, this is a long time to wait and a long to survive. pretty late in the game for them to make babies that then come and bolster the population, and then we have to wait another 15 years for those babies to become sexually mature. Once they are mature, they're aerial pairs like the surf smelt, so they do spawn multiple times in their lives, but this spawning is only going to occur every 1 to 5 years. For females, it's a little bit later, like 2 to 6 years. And like the Chinook, salmon, green sturgeon are anadromous, so they're born and they come back to spawn in freshwater systems. They're exposed to all those threats there. But green sturgeon are the most marine oriented of all the sturgeon. So when they're around 1 to 4 years old, they'll leave to the ocean, and that is where they spend the majority of their lives. So this long green occupancy period can expose them to some new threats, maybe bottom trawling, dredging, offshore ocean energy, wind projects. But it also poses this big question of what are they doing and where are they going? That's 6 years, we don't know. And green sturgeon are kind of an enigma. We don't know too much about them. We don't have any great, super accurate annual population size estimates. We don't know much about their habitat use or where they're distributed, especially for young fish. And there haven't been any specific mortality estimates done for some of the threats I mentioned, and also things like bycatch or poaching. So we have this dinosaur fish that is vulnerable, and we don't know too much about it. But we do know some things, of course. There are 2 genetically distinct population segments. They're going to range from the coastal waters of Mexico, but they're known only to spawn in California and Oregon rivers. So we have the northern DPS here in blue. They are characterized by fish that spawn in the Rogue River, Oregon, and the Klamath Eel River in California, and they are considered of conservation concern. And then we have the southern DPS here in orange, and they are known only to spawn in the Sacramento River, and also some 2 large tributaries of it, the Gila and the Feather. And they are listed as threatened under the Endangered Species Act. That is important. Both of these populations like to aggregate or hang out together in estuaries of non-natal rivers, so rivers they were born in, or some large West Coast bays. This has mostly been reported up in Washington and also in the San Francisco San Pablo Bay, where researchers there are theorizing this occurs when that bay or that estuary is about 2 degrees Celsius warmer than the adjacent ocean. So this likely is corresponding to some feeding behavior or some growing potential in these areas. And this red star here, this is us. This is my study location. We have got Humboldt Bay with the Mad River to the north and the Eel River to the south. Humboldt Bay is delineated into 3 main zones. We have North Bay, Entrance Bay, and South Bay. This is the 2nd largest embayment in the state of California. And maybe some of you are more familiar with this. We have some of the most productive and dynamic ecosystems in the state of California. We've got the intertidal mudflats, the eelgrass beds, the sand dunes, the coastal and freshwater marsh, and riparian forest. We also have several areas designated to protect wildlife. I boxed up this little one down here. This is the South Humboldt Bay State Marine Recreational Management Area. And lastly, the entirety of the Humboldt Bay is considered critical habitat for green sturgeon, but the Mad River and the Eel River are not. So I just want to zoom into North Humboldt Bay and point out 2 sites of interest for you. So this Red Star Sand Island, maybe you guys are familiar with this. This is a little man-made island in the northernmost region of this main central channel running up through North Humboldt Bay. And this is where word of mouth and public observations have recorded green sturgeon jumping around and hanging out here as early as the 80s and 60s. And also a study was conducted 20 years ago. They were looking to detect coho salmon, but incidentally reported green sturgeon spending a lot of time in this area. And then this yellow star down to the left, this is the site of a proposed wind power terminal plant. I do not know the current status or timing of this, but what they have proposed was construction that would take up aquaculture oyster farms in Humboldt Bay. Humboldt Bay produces over 70% of California's oysters. And the reason I'm showing you these 2 things is this is a lot of artificial infrastructure, either currently existing in the bay or proposed and maybe to come into the bay. And artificial infrastructure does hold the potential to interrupt some ecological processes. They could displace fish habitat and really just drives home how important it is to understand how sturgeon might be using these areas so we could learn how susceptible they might be to some of these threats. So I'm trying to document just the presence, distribution, and timing of great sturgeon in Humboldt County. So the who, what, where, why, all those questions. More specifically, I'd like to look into the extent they move into the bay, so how far are they coming in, and what are some specific aggregation hangout sites. And then I'd like to establish baseline data at that specific offshore wind power terminal site, and then evaluate how some individuals are moving and how that might relate to some environmental variables. So to do so, I have a bunch of methods I'm trying out, including acoustic telemetry, that's those visual surveys, citizen science, and pop-up satellite archival tags. So first I'm going to talk about acoustic telemetry. This is some of my juiciest results to share today. But what is acoustic telemetry? The basic underlying principle is sound waves. So you'll see, if you can make this out, there's a little tag in this chart. This is an acoustic tag. We can also pretend this is a sturgeon, they're tagged with the same type. And these tags, they're surgically implemented. I did not do any of this tagging myself, but there's a lot of agencies across the West Coast that have done this. And these tags have a battery life of around a decade. So any fish that has been tagged in the past decade could be incorporated into this study. What I am doing is I'm putting out these passive acoustic receivers. So these are stationary and stagnant in my study area. I deploy them in strategic ways to pick up fish. Because what happens is this fish will swim by, and every 30 to 90 seconds, that tag is sending out pings of sound waves for the receivers to record, which they can hear up to 700 meters away. And then they will log it with the date and time. And each tag has kind of like their own social security number, which I can then talk to a lot of people and figure out who it is. And then this active acoustic receiver up here, this is what I'm doing on the boat. This is basically just a handheld acoustic receiver that I can put in the water and listen in any area I can choose and just hope something swims by. So in a short season in 2024, I deployed 18 of those passive acoustic receivers. 17 were in the bay and 1 was in the Mad River. And just to break this down, so this is in South Bay. I have receivers at the top and the bottom of the 2 main channels, where the bottom is corresponding to that MPA I mentioned, and then Hoopton Slough. I also had a receiver in Elk River Slough, Freshwater Slough, and Mad River Slough, and this was to look into that objective of how far are they moving into the bay. But you'll see most of them are in North Humboldt Bay, a nice line leading up to Sand Island, as that's where I was expecting most sturgeon to be. I also tried to cover every entry and exit point, but you'll notice 2 white dots. I did lose these for a period of time, so I don't have any data to report on those today. And then the one in the Mad River, I put close to the mouth, lower down in the river. This was just looking to see if anything moved past on their way in or out. So of all these receivers, these ones remaining are the only receivers I got any detection, so pings from a tag in a fish on. So solely North Humboldt Bay. And what does this look like? So I had about 140,000 total detections. So again, detections are just the time the tag hitting the receiver. This could happen every 30 to 90 seconds, a lot of the time. It could just be sitting there being away for hours, which they did. And these were produced by 12 unique individuals. So in this image on the right, these green triangles, these are individual fish on the date they were first detected, at the receiver they were first detected at. So all of these fish were first detected on August 20th, which was the date I put the receiver out there. So this told me the fish were already there and I was late to the game. But they were also all detected at that Sand Island spot. The red triangles, these are the same fish, but the date they were last detected and the receiver they were last detected on. This orange triangle, this fish was first and last detected here. It likely left the bay, one of those spots I had a missing receiver and I didn't capture it. But I had fish leave as early as September 12th and as late as November 19th. The rest were pretty sporadic in between there. This here is just the raw number of pings from a tag, but it shows there was a clear peak in September that declined throughout the fall. And so who are these fish? Well, 11 of them were green sturgeon, and the 12th was actually a broad-nosed 7-gill shark. This was tagged in the Puget Sound in 2022 by Oregon State University. This is just a bonus in a study like this, but it's also really interesting. They're hanging out in the same areas around the same time as the sturgeon. And so there's a lot happening, but this is the movement of each fish between just my receivers. The main takeaway is just they're spending most of their time up here near Sand Island, definitely hanging out and aggregating here. It's hard to make out, but the ones that are blue hued colors, those are the northern DPS fish. I had 4 of them. They were either tagged in Washington or here in the Eel River. The red, orange, yellow colors, these are the southern DPS fish. I had 7 of these, and they were all tagged in the Sacramento River or the Yuba River, the tributary of the Sac. And this is a cool mix. I mean, it definitely highlights just how important this habitat is here for both population segments. And already mentioned, this fish was tagged in 2011, which was 14 years ago. And this was a big mom, so pretty fun. So what you're seeing now, this is another map of my receivers that are currently out there right now as we speak. The red dots are receivers that were out last year that I've put out again in the same place. The green triangles are new additions I've made. As you can see, I'm just loading up North Bay some more, trying to get some more info on what they're doing in these other 2 channels. I also had a receiver specifically out of that near that wind power terminal site that I put out in January. This was also acting as the just in case our sturgeon moving through in the winter when I don't think they'll be there. The answer was no tagged fish swam by that receiver at that location throughout April, from January to April. And this year I will also be incorporating the Eel River. And now I'm going to switch gears again and talk about visual surveys and citizen science. So in that same study period from August to December last year, I was conducting visual point count surveys. I was doing this weekly, so 1 week in North Bay, 1 week in South Bay, and alternating, where I would sit in these sites, which correspond with the bottom and the top of all the major channels in North and South Bay. And I would also sit there for 15 minutes and I would record some environmental variables like dissolved oxygen, water temperature, tidal stage and time of day. And most importantly, I would look and count for sturgeon out in the bay. I knew it was last. Also during the same period, I was distributing these flyers to solicit public observations of green sturgeon. These flyers had a QR code that linked to an iNaturalist project and also an email for people to send in their own sightings of these amazing fish flying around. And so this map, these are these 2 things combined. So these yellow diamonds are my visual surveys, the only locations I ever saw of green sturgeon, and I only ever saw them jumping. And the blue diamonds are all the locations I had public observations come in for 2024, which you can see they saw them in a lot of other places. Public observations were mostly jumping sturgeon, but I've had some people also see them under the water. A few people use drones, fish finding sonar, and there were a couple incidental bookings in the halibut fishery. And this is really cool. These are all methods I wasn't using to look for green sturgeon. So these absolutely enhanced my data set. And this here, this is the y-axis here. So this is the number of green sturgeon that were observed. And these are just dates. You can't really read that, but that's May 20th. And this is November 4th. The blue are the public observations, the yellow are mine. This is all in Humboldt Bay for 2024, except for the 2 rivers I pointed out. And so as you can see, the public was seeing green sturgeon as early as May. There definitely was a peak in July. This could be tourism, better weather, or maybe the halibut fishery. And I was just out there in the fall when the weather was a lot crummier. And I saw green sturgeon up until early October. And this is really cool. This absolutely highlights the importance of incorporating the community into your research. As you can see, they nearly doubled my data set. And this information absolutely informed how I set out those receivers in 2025. The public told me sturgeon were there as early as May, so I got those receivers out much earlier. They were out at the beginning of May. And the public also told me they were seeing green sturgeon in the Eel River, and so I started incorporating the Eel River this year as well. So lastly, pop-up satellite archival tags. So what are these tags? There's a lot going on here, but starting at the left, this fish has a little tag, well, it's actually a big tag on it. These are external tags, so most fish is tagged through a fin. For the sturgeon, it's going under those scoots. And you'll see this fish is moving horizontally across the water column, likely at different speeds. This fish is also moving up and down, likely at different temperatures, and throughout different times of the day. And these are all things these tags will report. And this fish over here, I can set a date that I want this tag to pop up. There's a little release mechanism that will send it floating to the surface, where it will transmit all that data to a satellite, which I can then download on my computer and make a map kind of like this of a track of what that fish was doing. So I got really excited to see some of this, but unfortunately I have not started doing this yet. Fishing is going to start this month, or hopefully, where I will go out and hook and line fish for green sturgeon in North Bay and administer these tags. But just to show you something, we have been practice tagging on white sturgeon in the Humboldt fish hatchery, trying to find the most efficient method to tag a beast of a fish like this. I think I got it, but we're gonna find out when we go out there. But this, you can kind of see the size of this tag here, this flotation mechanism, and it will pop off on the bottom, and I'm just attaching with monofilament line under those screws. So I just want to take a minute to bring back those objectives and just summarize all the information I just shared. So the presence, distribution, and timing, my acoustic receivers documented green sturgeon only in North Humboldt Bay. This was from August, started in August to December, but of course we know I was late to begin in August. These receivers detected 4 northern DPS fish and 7 southern population segment fish. My visual surveys, similar trend, only in North Humboldt Bay, really close to Sand Island and its main channel. But the public saw fish in a lot of other areas, including both of the rivers, the Mad River Slough and Plimpton Slough, which does indicate they are moving in pretty far to the bay, and in other regions that I did not capture. And the public told me this was happening as early as May. And then we know for any sturgeon to reach these North Bay sites, they would have to move past that proposed wind power terminal sites. And we also know this is spatially overlapping with those oyster aquaculture farms. It also seasonally is overlapping with aquaculture oyster harvest season, which can be characterized sometimes by increased vessel traffic and a little bit more disturbance. And then I showed a little bit of movement, but this is really going to be what my main goal is for 2025 with those satellite tags. And before I go, I just want to bring back these flyers and this QR code. My iNaturals project is called Humboldt County Green Sturgeon. I also have an email, pretty simple, greensturgeon@humboldt.edu. As you saw, these public observations were super helpful and really cool to have for my data. I also have this goal of just creating this network to continue on after I don't want to say I'm gone, but after I finish this project. So people can continue just reporting green sturgeon and monitoring them as a community. And the only information I'm looking for if you see a fish is the date and the location and how many you saw and any other intel you wish to provide. And I brought some of these flyers, I put them on the back counter if anybody wants to take one home to remember. And yeah, so that is the end of my sturgeon spiel. And I'm just going to wrap this up really quick and bring back the 3 fish we talked about. So we put a lot of information on you guys, but there's one common theme I want to point out is these are all data limited species. So Rachel told us that this is the first study in 30 years looking at juvenile Chinook salmon in the Eel River estuary. Daniel let us know that for surf smelt, he's the first study to incorporate those biological indicators. And for green sturgeon, mine is kind of the first direct study in Humboldt Bay on these population dynamics as well. So this, given that they are so data limited, it proves the importance of monitoring native species that are again of important cultural, economic, and ecological value to the tribes in this area. And we really hope that these projects can enhance this monitoring, but also enhance tribal stewardship through co-creation of knowledge and co-creation of these new methodologies for monitoring. And that is all we have. Lots of people think I'm not going to read the whole list, but it takes a village. And there's 3 of us here, so there's more that are on this list. But that is it for us, and I will bring back up these 2 so we can answer questions. Thank you. Yes, question.
I have a question about the timing of the sturgeon coming into the bay and what are they coming in?
So, likely for feeding up in North Humboldt Bay, there's a lot of crustaceans, a lot of little crabs, little skunks up there. And the timing, so they're coming in May, maybe about October. It is theorized that this exit period corresponds to that onset of those heavy winter fall rains. So it could be there's something indicating a change in some sort of water quality parameter or that they're leaving, or maybe it's just a drop off in that food productivity. But that's kind of hard to say without doing a diet study, which I simply can't do more. That answers the question.
Yes, but can I ask Daniel the same question about surf smell? And there, you mentioned that you sampled them on the beach. So what is the extent out into the surf zone that you would find surf smell? How far out beyond the first breaker to
We can see them through birds. We have an idea that they're up to a quarter mile north from the Pelicans. And yeah, certain sites. We'll see the signs, we'll move because they're not coming on shore, and then sometimes like 2 hours later, or at the end of our sampling event, we'll see them come on shore that same area. And catch them as well.
Wow.
With your green sturgeon project, was there a way that you were validating science green sturgeons as species around here?
So no, white sturgeon are very uncommon in this area. I don't want to say it doesn't happen because apparently it's happening in but it is so infrequent that I'm not. And there's also I have some educational materials on the iNaturalist. I have my examples and my pictures to give examples of them. But most of the time it's the green sturgeon side of confidence, and I don't do too much of my background check.
Question for Daniel, why are you sampling the northern sites instead of based on southern sites?
So we're sampling the northern site because we're trying to look at the effects of the lunar phases, and we added additional sites, so we couldn't do them on consecutive days because we're looking at so new set. Sure.
what is the relation and lunar phase?
Uh, the, the springtime, we had a new design, but based on the days that we have so far, been more productive.
So can I continue?
Yeah, we have.
So that means the high tides are higher.
Yes.
And spawning might be higher on the beach. What is the relationship between the physical, biological facet of the spawning and the magnitude of the high tide?
So on recruitment, it needs to be a certain percent humid, and like the temperature needs to be under 20 degrees. So it's it could be just that there's a certain stretches of the beach that are accessible, and they're gonna be like stronger like stronger tides. So maybe they have more access to the beach, or like just different pools on them, different center.
I also have a question for Daniel.
Yeah.
Do you have any potential thoughts or ideas?
There's really on them, and so it's kind of like scout, like going out there checking out if it's ideal spawning habitat, and kind of okay, send the males and you get kicked off, and then once they know it's ideal spawning habitat, then females can come in and spawn. And the literature suggests that outside of For some reason, when they spawn, they don't need as many females.
So would it be possible if you're using a different sampling here that go farther out? You might get more of that 50-50 sex ratio? Is that kind of what you're?
But yeah, for sure, potentially, so we're limited because we sample with an A-frame and in waders, so you can only go up to our waist about, and generally when we catch them, they're all below our knees, but yeah, that limits us.
Just to,
So we decided to use their traditional gear, because they wanted to kind of sample. Yeah, they're gonna continue this on and they wanted to do it with their methodologies, and so, we used their nets. Mhm.
yeah, it's wrong, yes. So you brought up, you had some results with regards to water temperature, were there any results with other environmental factors?
I have not explored that too much yet. Yeah, the 2024 season was sort of like a preliminary, trying to get data, figure out where they are occurring. But that will be something I'm looking at for final.
And you said you were considering water temperature, salt, oxygen, salinity, was there any thought on including like turbidity or anything else, or were those the main three?
I did try to include turbidity last season, but it's a really difficult parameter to add to measure accurately, so I decided to drop that one. But there is some it might be a helpful parameter, in terms of like predation or the higher turbidity. Yeah, if you have an experience, but it was difficult to get accurately out there.
And you just mentioned predation, and it is an alternative. Hypothesis is,
That happens. Out there, a lot of bird predators. But another aspect of my monitoring the pipe minnow and see how far they can travel into the estuary. Because we are seeing some larger fish at those topmost sites, which would mean that there's potentially more predation up top.
yeah, have you talked to Dan Barton? About, not, what's that?
About bird predation.
About bird predation on Sand Island with the number of, okay. We're not tagging these fish though, so we'll never see them. Right, but it still offers you insight into the volume of predation by cormorants and terns on San Juan that was unappreciated.
Yeah, I'm sure it's happening down there. There's lots of comments.
Yeah. So for you again, for how many satellite tags are you expecting to have, and what is sort of the time frame you're expecting to attempt to have them on the fish board?
Good question. So I have 8 satellite tags, they were donated to the project, so that's what I have, like, that's it, no other reason. They can stay on the fish for up to a year, so I might try that, but that's a long time to wait.
So maybe you the ones that are just, you can get it once they pop up and reach the surface.
A little bit of both. So when they're at the surface, they transit data, it will be bin data, so it'll be like every like hours for bin or like days for bin, so you could also suffice. Yeah, it's gonna be a learning experience. I do have some collaborators that have handled green sturgeon and captured and tagged them before, but on the fishing end, just asking the fishermen. Okay. Tips and trips are welcome if you've have experience fishing for a monster, really angry shark and pointing sturgeon. I'm here. Cool. Any other questions? Great. Well, thank you guys so much.
And hey, everybody, our next Wednesday, first of the month, speakers will be Jennifer Cole, the executive director. Waterkeeper, and her associate Sylvia, they'll be talking about here at Marsh, past, present, and future. So if you make it to that, that'd be super. Pass on the word. And thank you so much for being here. Thank you for